Hello everyone, I am Jahnavi. Today we are going to discuss about various cloning enzymes. Gene cloning is the process in which a gene of interest is located and copied out of all the DNA extracted from an organism. There are 8 types of cloning enzymes and they are nucleases, polymerases, ligases, transferases, kinases, DNases, RNases and phosphatases. Let's now look at a brief discussion regarding nucleases. Any enzymes degrading nucleic acids are known as nucleases. There are 2 types of nucleases, exonucleases and endonucleases. These nucleases are responsible for cleaving the phosphodiester bonds between the nucleotides of the nucleic acids. Defects in certain nucleases can cause genetic instability or immunodeficiency. Nucleases are also extensively used in molecular cloning. There are 2 types of nucleases, exonuclease and endonuclease. Let's now discuss about exonucleases. Exonucleases removes nucleotides one at a time from a DNA molecule from 3' to 5' ends of DNA and RNA chains, whereas endonucleases breaks the phosphodiester bonds within the DNA molecule. Exonucleases performs their activity outside the DNA, that is, towards the ends of the DNA, whereas endonucleases cuts or cleaves a fragment in between the 5' and 3' ends. Examples of exonucleases are exonuclease 3 and exonuclease 1. Exonuclease 3 works on dsDNA, that is double-stranded DNA, whereas exonuclease 1 works only on single-stranded DNA. Examples of endonucleases. DNase 1. DNase 1 work for both single-stranded DNA as well as the double-stranded DNA. For double-stranded DNA, manganese ions are nicked to the both strands, whereas for the single-stranded DNA, magnesium ions are nicked to the single strand. You can now look at the activity of nuclease enzymes. The first picture describes the activity of exonuclease cutting from the edges. The second picture describes the activity of endonuclease cleaving in between the strands. The second enzyme is polymerases. DNA polymerases generally occur during DNA replication or cell division or distribution of DNA and for maintenance of equal amount of DNA between the offsprings and mitosis. There are different types of DNA polymerases. DNA polymerase 1, 2, 3, 4 and 5. DNA polymerase is responsible for the process of DNA replication, during which a single-stranded DNA molecule is copied into 2 identical DNA molecules by addition of nucleotides at 3' end of the DNA, and by doing this, many methods such as PCR and other can be achieved. Our next enzyme is ligases. DNA ligases play an essential role in maintaining genomic integrity by joining breaks in the phosphodiester backbone of DNA that occurs during replication and recombination. You can now look at the picture attached to understand the activity of ligases. There are 4 types of DNA ligases: bacteriophage T4 DNA ligase, E. coli DNA ligase, Taq DNA ligase, and T4 DNA ligase. Our next enzyme is transferases. Transferases are the enzymes that catalyzes the transfer of specific functional group from one molecule to other. Activity of transferase is explained in the figure where aspartate is transferred and carbonyl aspartate is formed. Our next enzyme is kinase. Kinases transfers gamma phosphate of ATP to 5' end of polynucleotides. Gamma phosphate group is the primary phosphate group on the ATP molecule, which is hydrolyzed when the energy is needed to drive anabolic reactions. These kinases are useful for preparing DNA fragments for ligation. These are also useful for radiolabeling DNA fragments using gamma ATP as a phosphate donor. As shown in the figure, a protein is converted into a phosphorylated protein by the action of protein kinase, which includes formation of ADP from ATP. Our next enzyme is DNase. DNases are the enzymes which hydrolyzes DNA by cutting the PO bond, that is phosphate and oxygen bond attached to the deoxyribose of 5' carbon. These are also known as deoxyribonucleases. You can observe the figure to know the activity of DNases. The next enzyme is RNases. Enzymes which hydrolyze RNA by cutting the PO ester bond attached to the ribose 5' carbon. are known as RNases. It represents typical example of acid-base catalysis. These are also called as ribonucleases. The last enzyme is phosphatases. Phosphate group is removed from the substrate by the action of these phosphatases. Observing the figure given, we can say that the phosphate group is removed from the substrate. Thank you for watching. Kindly do like, comment and share.